Professor Rasmus Nielsen. Professor Nielsen got his PhD at Berkeley, and he has been on faculty at Cornell and University of Copenhagen. He's now again at Berkeley, and he has, through his career, moved from long-term evolution in phylogenetics to short-term genetics, and mixed the two in his most recent work, combining evolution and genetic work. And he's going to give us a talk on adaptation of the human genome and finding evolutionary adaptation through a number of innovative methods. Those of you who have worked like I have in phylogeny will remember the panel package and a number of other very important tools. I think we have just as much to expect from today's talk. So please join me in welcoming Dr. Rasmus Nielsen. And to thank him for attending APBC and giving us his talk. There is a small presentation on the part of the APBC organizing committee.
Thank you very much.
Oh, thanks.
Appreciate it. Thank you for the introduction, and also let me just thank the organizers for inviting me. It's been a pleasure already to be at this meeting and heard a number of great talks. So it's exciting. Hopefully, I'll have the opportunity to come back to this meeting some other time. Okay, so outline of my talk is two parts. There'll be a part that I hope you'll find exciting, and then the boring part after that. So the more exciting part is the first part, where we'll talk about two examples of human adaptation we've been working on. And what I want to do in these two examples is illustrate the principle that you can use evolutionary inferences to learn something about function. So even if you're not an evolutionary biologist, and even if you don't care about evolution, you only care about functional aspects of genomics and biology, it might be useful to know something about evolution because it actually can help guide you make functional inferences. So I will illustrate that with these two examples. And then that will lead me to talk about something else we've been interested in for the past few years, which is some theory on what is called admixture tracts or admixture fragments. We developed some theory, population theory for that that I'd like to share with you. And I think I will motivate why we're doing that also in terms of these two studies. One thing that we've been doing a lot over the past 10 years is to study human migrations and human demography using ancient DNA in collaboration with Eske Willeslev's lab at the University of Copenhagen, also in collaboration with Svante Pabo at the Max Planck Institute, and also looking at modern DNA sequences. We've done a number of studies where we're trying to elucidate the patterns by which modern humans came out of Africa somewhere maybe 60, 70,000 years ago, and then spread out to the rest of the world. For example, we worked a lot on how the Americas was colonized. We now know from our studies that Native Americans and East Siberian population and most East Asians, they diverged from each other about 23,000 years ago. And then after that, Native Americans merged with another population from Siberia, the ancestors, and then migrated into the Americas. And you start finding archaeological evidence of Native Americans in the Americas close to 15,000 years ago. We worked on that. We also worked on Australia, Europe, and other places in the world. And many other groups have been interested in these same problems. I think now we're getting a good idea, a good picture of how the world was colonized by human migration. That's not really the topic of today's talk. What we also have been interested in is what then happened when humans migrated into different parts of the world. They came from areas in Africa that was tropical or subtropical, with not very much variability in climate throughout the year, and then they conquered all these different climatic zones and different environments. For example, they adapted to life in the high Arctic, they adapted to life in the Tibetan plateau, in the high altitude there, and to many other different environments. And one of the things we've been interested in is what happened biologically as humans moved out of Africa and adapted to all these different environments. And if you look at humans, an enormous amount of cultural adaptations, obviously, to different environments. But we also think that there's a great deal of biological adaptation. Literature, the anthropological literature, are full of claims of this. But in humans, it's very difficult to do controlled studies where you really test rigorously what is a genetic and evolutionary adaptation, and what is really environmentally driven. So we don't really know in many cases how much of this is actually genetics. And then that's one of the things that we're interested in, trying to figure out what are genetic adaptations humans have to different environments. And just to motivate a little bit further, why we're interested in that? Well, because it might often lead us to important functional differences. Well, we're interested in also because, of course, it's interesting from an evolutionary standpoint. We want to understand where we all come from and how we have adapted to environments. But it's also important for understanding functional differences between humans. Why do we react different to different diets? Why might we have different responses to different diseases? Many of you are bioinformaticists, so you will use evolutionary inferences in your daily work to make inferences about what might be functional. One of the most common tools, of course, in bioinformatics is that you take a set of aligned sequences and then you ask, what's conserved? Where do you have regions in these aligned sequences where all the DNA from all the different individuals or different species is the same? If it's highly conserved, you infer it's likely to be functional. It's one of the most basic tools we have in bioinformatics. Now, that kind of inference tells you something about what's functional and the same among all individuals. But often a much more important biological question is, what are the functional differences that make us different from each other, that make us respond differently to different diseases, respond differently to different diets, and so on? If you're interested in that question, what you have to look at is human adaptation, the parts of the genome where we're adapted to different environments. We're using to detect adaptation is to finding the footprints of natural selection in the genome. And so very briefly, what are we looking for? Well, you can imagine that you have a population initially like this. These are individuals in the population. Then you have a mutation, so this individual is now different from the other one. Now, if that mutation confers a fitness advantage to the individuals carrying it, in other words, if the individuals carrying this mutation in average leave more offspring in the next generation, then with some probability, eventually, the body in the population will carry that mutation, and we talk about positive selection. And so that's what we often are looking for. We want to find these footprints of positive selection, of examples of where selection has favored specific mutations, where selection has driven change in the population. I'm going to talk about this Tibetan altitude adaptation. This is a joint project we did together with BGI, Virginia Genomics Institute, about 6-7 years ago. Now, the plateau is a height of about an altitude of about 4,000 meters. So that environment is quite hypoxic, meaning there's low oxygen. The pressure of oxygen at the Tibetan plateau is only about 60% of what it is at sea level. So if we go up, most of us, if we go up in that altitude, we will suffer from oxygen deprivation. Now, then there will be a physiological response to that. We'll start acclimatizing. And one of the ways we'll produce many more red blood cells. That's a primary way we'll be trying to acclimatize. So we can now, because we have more red blood cells, transport more oxygen in our blood and therefore better deal with the situation that there's less oxygen in the air, or the particle pressure of oxygen is lower. Now that you think is a good thing, but not necessarily, because it has a side effect that the viscosity of the blood. The blood becomes thicker. The chance of stroke. And it has also other negative side effects. It's associated with increased hypertension, preeclampsia in pregnant women, and so on. So there's strong negative fitness effects of the normal physiological response to low oxygen at high altitude. Now, Tibetans are special in the sense that it's been described already that they have a slightly different response. They don't produce quite as many red blood cells in high altitude. Okay, so they have genetic adaptations that allows them to avoid these negative fitness consequences of producing too many red blood cells. And so what BGI and us, what we're interested in was to identify what are the regions in the genome in the Tibetans that allow them to do that. And the tool that we used for that was not to do a big genome-wide association mapping study. The tool we used for that was to try to identify what are the regions in the genome where there's been natural selection, positive selection, specifically in the Tibetans. So we did one of the first exome sequencing studies that were ever done. It was published in 2010. But back then, there hadn't been many exome sequencing studies. And we sequenced DNA 50 Tibetans, and from 40 Han Chinese, or BDI did that. And this is basically the result of it. This is a heat map of the allele frequencies. So SNPs up here, those are SNPs that have a high allele frequency in Han Chinese and in Tibetans. And SNPs down here are SNPs that have a low frequency in Tibetans and Han Chinese. So we learn a couple of things from this. First of all, allele frequencies in Han Chinese and Tibetans are quite closely correlated. So that means that these are closely related populations. They have very similar allele frequencies. But we can also see that there's some outliers. In particular, there's one strong outlier. We see these two SNPs here that have a high derived allele frequency in Tibetans and relatively low allele frequency in Hans. They're both in a gene called EPA12. And we had a statistic that we derived that basically measures something about how much positive selection has been driving changes in allele frequencies, specifically in the Tibetan population, by comparing them to the Hans and also to another European output that we used. And using this statistic, also EPAS1 as a gene stood up very, very strongly. It's an extreme outlier. And we can also assign some p-values to it, and we do that using various simulation procedures where the p-values, we could only estimate that the p-values is something that's much less than 10 to the minus 6, but we couldn't do enough simulations to really figure out what the p-value was. It was very, very small p-value associated with the hypothesis that it could just be genetic drift that caused the change in EPAS1. You can reject that null hypothesis with strong statistical confidence. So EPAS1, there's something going on in this particular gene. And then we could well, gene associated with the phenotypic difference that we're interested in, which was red blood cell concentration, or a proxy for that, mean hemoglobin concentration. Now, we looked at a particular variant where there's a CD polymorphism, where the frequency of G on Tibetans is 87%, and among Han Chinese it's 8%. So a huge allele frequency difference. That was our strongest hit. And if we look at mean hemoglobin concentration, we can see that if you're homozygous for the GT phenotype, you have a much smaller mean hemoglobin concentration than if you're heterozygous or homozygous for the other type. So we can explain much of the variance among individuals in mean hemoglobin concentration, red blood cell concentration, by association with this variant. So we have found the gene that seems to be associated with the lower production at high altitude of red blood cells in Tibetans. What does it do? Well, it's a transcription factor. It regulates the response to hypoxia. It's already known to be important in fetal heart development and also in the regulation of blood cell production. And there have been already found allelic variants associated with chronic mountain sickness. It's a gene that sits very high up in the signaling pathway that's called the response to hypoxia pathway. So when you're deprived of oxygen, it's one of the first genes that'll become activate a number of other genes in a cascade that will, for example, lead to the increased production of red blood cells. So it's a very, very good candidate gene that we found here. Okay, so that was exciting part. And we kept working on this for the next couple of years because we're mostly doing mathematical and computational research. We like to model what we see, and we like to establish models of the way natural selection works. We like to be able to describe everything that's going on in the genomic region around EPAS1 and estimate things like the time at which selection started acting, how much selection, what's called the selection coefficient, acting on the mutation, and so on. And the problem we had when we did that was that none of the models we made fit the data very well. They're all quite poor models. And the problem was that the differences between the haplotypes that you see in Tibet and the ones you see among the Han Chinese was just too big. All right, so this illustrates that. So this is Tibetans here and the Han Chinese here. Each row here is a haplotype, and each column is a particular SNP. And you can, so white would indicate the ancestral type and black the derived type. And you can kind of just, if you just squint and see that there's two very different haplotypes here that differ by a lot of SNPs. And they actually differ by many more SNPs than you would expect if selection is just acting on a single genetic variant that's causal. So that could be explanation from this. Well, maybe there are multiple SNPs that selection is acting on at the same time. And there's some kind of epistasis. Is there some kind of epistasis that can create these longer haplotypes? But a model like that also didn't fit the data very well. We started to think about maybe there's a haplotype coming from somewhere else into the Tibetan population. But it turns out that the Tibetan population, the haplotype that we see there, doesn't exist in any other human populations. Well, it exists in closely related populations in Mongolia and among some people in Nepal and so on, but doesn't really exist outside the Himalayas. And so it doesn't really seem like it was suddenly, a sequence that came in from Africa and something. So we thought, oh, maybe it is Neanderthal integrates DNA, but it doesn't match the Neanderthal DNA either. Then there's, and many of you will be familiar with the fact that there's another group of hominins that when Svante Pääbo, he sequenced Neanderthal DNA, shortly after he found also this other group of hominins that are related to Neanderthals, but not Neanderthals. They're called Denisovans. They're known only from a tooth and two teeth and a little bone fragment from a finger from a cave in south central Siberia near a place called Denisova, hence the name Denisovans. And when we compare to the genome sequence that Svante Pääbo had generated for these Denisovans, Svante Pääbo, and this is the little finger bone that led to the discovery of these of the Denisovans. When we looked at that sequence, we found there was a complete match to the Denisovans. All right, the Denisovan sequence, an almost complete match between the Denisovan sequence and the Tibetan sequence. And then we could do various other analysis where we could show statistically that getting this much similarity in such a long region in the genome could only have happened given what we know about the divergence time between Denisovans and humans if there's been integration, the transfer of DNA by interbreeding between humans and Denisovans. So this is a large fragment that's been transferred by interbreeding from Denisovans into the ancestors of the Tibetans. There's a haplotype network that illustrates the relationship between different human haplotypes and then Denisovans. And what you can see here, these are each pie here is a haplotype, and the color scheme indicates different human populations. So, for example, you have the yellow here, that's Han Chinese individuals. And there are different African populations here in blue. And the Denisovan haplotype sits down here, and here you have the Tibetan haplotype that's related to the Denisovan one. So the Tibetan haplotype differ from the nearest human haplotype by 40 mutations. And it differs by the Denisovan from only 9 mutations. And the Denisovan haplotype differ from other human haplotype, the close one, by 35 mutations. So the Denisovan is actually closer to any existing modern human sequences, except for the Tibetans, than that Tibetan sequences. And that's exactly what you would expect if there's been integration. Why is that? That's because this is an ancient sequence that might be 50,000 years old. So it has a shorter amount of time to accumulate mutations. So that should be closer to other human sequences than the Tibetan is if it's actually integration. So that's one of several lines of evidence that we use to establish the fact that, in fact, this is an example of integration from Denisovans into humans. What this led us to, so there are other examples of integration in modern humans, particularly from Neanderthals, that have transferred adaptive genes from these archaic species into modern humans. And it's leading us to a picture of human evolution that shows that integration really is much more important than we previously thought as humans adapting to new environments. So humans in these about 60,000 years have adapted to all these different environments. And it seems like one of the keys of how we could adapt to all these different environments is because there was already other hominids in these environments that were already adapted to those environments. And by integrating with those individuals from other species, we could get up some of the key genes necessary for survival in those environments. Okay, so it's slightly changing our view of human evolution, I would say. Okay. My next example, which is an example based on Inuits. So Inuit are the indigenous people that live around the Arctic, in the Americas, and in Greenland, people that previously were called Eskimos. And we're interested in them for two different reasons. First of all, they've been hypothesized to have various adaptations to cold, right? They live in a very cold environment, and they're supposed to have adaptations to that, although nobody have ever shown that they're actually genetic adaptations, that it's not just a kind of acclimatization. All right. We're also interested in them for another reason, that is that they have a very, very specialized diet. And the specialization in this diet has been used to make inferences about what might be healthy and what might not be healthy to eat. In particular, they have a diet that is very, very rich in fats, but also to some degree protein from marine mammals and fish. So they eat a high their diet has a very high concentration of certain fatty acids, certain polyunsaturated fatty acids that you find in marine mammals and fish. So when I, from my office, go down to Starbucks to buy a cup of coffee, there will be a little option on the lunch, lunch boxes that is the omega-3 lunch that they have. When you go to the supermarket, you might find milk that's been enriched with omega-3 polyunsaturated fatty acids. It's a special kind of polyunsaturated fatty acids that's very common in marine mammals and in fish. They're supposed to be healthy for you and protective against cardiovascular disease. Why is it supposed to be that? Well, the first studies arguing that it's healthy were studies studying the Inuit. They realized that the Inuit have this diet that's really rich in fat. They eat all of this fat. But on that traditional diet, they were perfectly healthy and suffered from a very, very low rate. The incidence rate of cardiovascular disease was extremely low. They did not suffer from many of the diseases you would expect people to have if they eat, if they have a diet that's extremely rich in fat. And so what the research pointed to was, well, the kind of fat they're eating is really rich in these omega-3 polyunsaturated fatty acids. So perhaps that's protective against heart disease. And that led to 40, 50 years of research and enthusiasm for the possible health benefits of these omega-3s. So interested in, so this extreme diet, has that changed that genetic makeup? Is there any effect genetically in the Inuit? And could perhaps the fact that they have this diet, could that have had an influence on their physiological response through genetic mechanisms? We worked together with epidemiologists that have been working in Greenland for a long time, and we put some money together, a couple of me and a couple of my former students, to genotype, using a genotyping chip, 2,500 Inuits in Greenland, for which there was extensive phenotypic data available for them. One of the first things we wanted to look at was true diabetes, because now we had the data. We knew that Inuits on a modern diet is one of the populations in the world that has the highest rate of type 2 diabetes. So we wanted to first ask, before we went back to this question about adaptation, do you have any spatial mutations for that? And I don't know how familiar you are with the genetic base of type 2 diabetes, but hundreds of millions of dollars have been spent on trying to find that genetic basis. And basically, today, I would say that the results have overall been disappointing. There are no genetic variants with a strong effect on type 2 diabetes. We have many variants that contribute a little bit, maybe with a relative risk of 1.2 at most, and that will not then be very common mutations. So it's a highly polygenic trait, and most of the genetic variants has not yet to be discovered. It's one of these traits with a lot of missing heritability. That's because people have been studying primarily Europeans, because the story in the Inuits is quite different. So this is a Manhattan plot. You have the different chromosomes in the genome here, and this minus log 10 p-value for an association test between type 2 diabetes and particular genetic variants. And you can see there's one particular position in the genome where we get this very strongly significant result. It's a gene called TBC1D4 that really gets a high degree of the genetic variants in the Inuit population. There's the relative risk associated with this variant is larger than 10. Okay, it's almost Mendelian. If you're 60 years old, you're homozygous for this variant, then you almost certainly have to have type 2 diabetes. It's almost like a Mendelian in the Inuit population. It's a completely different story than what you have seen in other populations. And we think that perhaps it has to do also with the diet, because in the traditional diets, they actually, even though I say it's Mendelian, they actually don't tend to get type 2 diabetes with that mutation. It's only on a modern diet. And that's perhaps what we think is going on, is that they have a diet that's very low in carbohydrates. It's highly hypoglycemic. They get no sugar in the traditional diet. And that mutation, what that it does is it modulates the uptake of the cell of glucose. And we think that mutation perhaps could be adapted under that traditional diet that's very low in sugar, but very bad for you on the modern diet. What happens is that the mutations like that mutation, in the traditional lifestyle, are beneficial for you. But then in the modern lifestyle, when suddenly you change and you have a lot of sugar in your diet, it's very bad for you. All right. So now on to the selection scan. So now we did the same thing Inuit, as we've done for the Tibetans, we're trying to find particular positions in the genome associated with positive selection. And this is again something similar to a Manhattan plot where you have different positions in the genome here, and then you have a test statistic that tells you something about the strength of the evidence favoring that there was positive selection, particularly in the Inuit, in a particular position in the genome. And you see there's a couple of sort of extreme peaks here, in particular one in a region of some genes that are called FATS genes, FATS1, FATS2, and FATS3. What these genes do, where you find all this evidence of natural selection, those are the genes that are crucial in the synthesis of polyunsaturated fatty acids, in the omega-3s that we talked about before. And in fact, what they do is, we could go back and when we well, maybe I'll explain that here. So this is the synthesis pathway of omega-6s and the synthesis pathway of omega-3s. And both of these gene FATS1 and FATS2, they both contribute to these synthesis pathways. And what the kind of omega fatty acids that you would get from a fish diet are these omega-3s, the 25s. And from a meat diet, you would tend to get these polyunsaturated fatty acids. And what we can do is in the individuals, in the Inuit, we can measure in the cell membranes the composition of different fatty acids. And what we can show is that this particular mutation, this mutation that we find selection acting on, is downregulating that particular step. So you produce yourself much less of these fatty acids that they get from their diets. So what we think is going on is that they have this diet that's very rich in both protein and also in protein and fatty acids, polyunsaturated fatty acids, both from meat and from fish. And because of that, they downregulate the genes that produce those very same polyunsaturated fatty acids. So the Inuit have particular genetic adaptations to a diet that's rich in polyunsaturated fatty acids. Because they eat more of those on a traditional diet, they produce themselves physiologically fewer of them. So there are a number of downstream consequences of that. And there's an extremely strong association with LDL cholesterol. And it's again one of the strongest associations with LDL cholesterol ever found. So if you re-regulate the production of polyunsaturated fatty acids, there's a strong effect on cholesterol, that we see a very strong effect in the Inuit of that. But also on other traits, such as weight and in particular height. If this mutation that re-regulates the production of polyunsaturated fatty acids, if you're homozygous for that, you're on average, as an Inuit, 2 centimeters shorter than if you're homozygous for the other type. So that's the common mutation ever found with the strongest effect on height ever found in any population. All right, so how does this work? We're a little bit puzzled by that. How you change the production of a fatty acid and then suddenly you have this enormously strong effect on height. That work. Well, it turns out that relationship, a well-known relationship between release of growth hormone and the fatty acid profile. So, for example, chicken breeders, animal breeders, they're clever people and they will figure out how can you make the biggest chicken. And what they figure out is that if you, instead of having vegetable oils, add fish oils to the feed for the chicken, then the chicken will become much larger. And the reason why they'll do that is that they will produce much more growth hormone. The blood concentration of growth hormone will be larger, and the chicken will become larger. And something similar seems to be going on in humans as well. That if you switch to a diet that's, if you have more certain polyunsaturated fatty acids, you will release more growth hormone and you will become larger. So because of the Inuits genetically produce less of it, if you have that mutation that allows you, that will make you produce less polyunsaturated fatty acids, you'll become shorter. All right. We could also validate several of these results. We could validate them in other cohorts. For example, we see a similar effect on height in Europeans, but it's just that this mutation is extremely rare in Europeans. People have not noticed it before. It only has a frequency about 1 or 2% in Europeans. So all the large genome-wide association mapping studies looking at height, they haven't had it as one of the most important genetic variants because of its low frequency in Europeans. But because of its selection as favored in the Inuit, we can see it in the Inuit and see the strong effect it has on height in the Inuit. All right, so to sum up the conclusion of this part of this topic, we think there's been a change in diet in the Inuit originally to a diet that's really rich in meat, and particular meat and from fish and from marine mammals. Selection on these fat genes that are responsible for polyunsaturated fatty acids, which leads to a modification of the fatty acid profile. That has a lot of downstream consequences. LDL cholesterol, it also has, and I didn't talk about that, a strong effect on fasting insulin levels, hormone control, to reduce height, because all these downstream effects. Now, another important point from this is, of course, that we know now has special genetic adaptations to a diet rich in polyunsaturated fatty acids, particularly high concentration of omega-3 polyunsaturated acids that other populations don't have. This is a mutation that in Admixture, Inuit has a frequency of maybe 98%, but in Europeans a frequency of maybe 1 or 2%. So it's clear that we lessons from the Inuits regarding polyunsaturated fatty acids and what might be healthy for you cannot be extrapolated to other populations. So the idea that just because the Inuit are very healthy on a diet that's very rich in fat from marine mammals, that doesn't necessarily mean that it's healthy for the rest of us to eat a large amount of omega-3 fatty acids of fish all the time. And that's a bit important because recently there's been a lot of studies that have questioned, is it really in fact healthy for you to eat omega-3 fatty acids? There's some large epidemiological studies that have claimed there's no health benefit of it. Still a controversial area. Okay, so this is all published. Let me just show a few unpublished results, particularly the second peak here. We didn't really have time to talk much about it in the first publication we made on this, but the second peak here is also interesting. It lands in this TBX15 ROS2 cluster of genes. A little bit about it. It is associated with processes, but one of them is the production and regulation of brown adipocytes, brown fat cells. So if you change the expression of TBX15, you're more likely to get more brown fat cells. Why is that interesting in this context? Well, brown fat cells are thermogenic. They generate heat. So we think that this might be important on the underlying mechanism here in terms of evolution might be selection relating to adaptation to cold, to the production of a higher concentration of brown fat cells. It's also interesting for another reason, because it turns out that fragment TBX is also integrated, either from Denisovans or from Neanderthals. It's a bit, that region, Denisovans and Neanderthals are quite similar to each other, so it's hard with great certainty to establish which one is from, but it's closer to Denisovans than it is to Neanderthals. And this is a haplotype network where you have Neanderthals here and Denisovans, and this is that integrated haplotype. Okay, so that is actually also a reasonably high frequency in some other populations, for particular other Native American populations and also some East Asians. Okay, and it's clearly integrated from either Denisovans and from Neanderthals. All right, so this is another example where we're looking at adaptation, and again we find integration, and perhaps that the cold adaptation, human moved out of Africa, they had to adapt to colder environments as well. And the genetic makeup that they needed to be able to do that, perhaps they got that also from integration with Neanderthals or other hominids. Okay, so we keep running into, when we do this work, integration again and again and again. And so that led us to increased interest in the theory of how admixture works. It's very related to what's known as ancestry painting that you might be familiar with. So this is from 23andMe's website, where they give an example of ancestry painting. So these are different chromosomes in an individual, chromosome 1 and 2 and so on. And then you could take that individual and then use large reference databases of allele frequencies in different populations. And then you could say which part of that individual's genome is, say, European or African or Asian. So you can see this is somebody that self-identifies as African American and has a large proportion of African genetic background, but also some European genetic background. And if you look at different parts of the genome, you can find these tracts. That's what we call admixture or integration tracts. And we're interested in the mathematical theory of that. How do these tracts evolve And can we use that to say something about when admixture happened? And can we also say something, for example, about if natural selection has happened? So therefore, we have been doing quite a lot of work over the past few years on the theory of these admixture tracts. And one of the first things to notice is that what almost all studies assume is that you can describe the process of how you lay down these admixture tracts. You can describe it as a Markov chain. So 23andMe and everybody else doing this kind of ancestry painting, they use a hidden Markov model to do the ancestry painting. So there's an assumption of a Markovian process governing the distribution of these ancestry tracts. So that was one of the things we were interested in. Is that a realistic assumption? Part of why we're interested in that is we know that the parameters of this HMM is very important for this ancestry painting. So perhaps if it's not even a Markov chain, we're making some wrong inferences once in a while by assuming by using an HMM. What we're interested in is this process of how these admixture tracts, how they evolve. So you can imagine now that you have a first generation admixed individual, like an F1. And imagine, for example, here the red chromosomes. So these are two chromosomes, one from your mother, one from your father. You can imagine your mother is human and has a red tract, and your father is, say, Neanderthal and has a blue tract. So in the first generation, after admixture, this is how you look genetically. Now, if you then cross into humans, you keep breeding within humans. There's no more integration. Neanderthals die out, and now it's all just breeding within humans. You will start to break up these chromosomes due to recombination. So now this chromosome will be shorter. After one generation, after 5 generations, you might have broken up smaller tracts like this, and 100 generations, you might have smaller tracts like in the fresh DNA. So one of the important things here to realize is that the longer time has passed, the shorter these integration fragments will be. And so we can use that to, for example, infer when did the integration happen by looking at the length distribution of these admixture tracts. Okay, we did some years ago, this is all the way back to 2009, we could, if you just assume that you have these admixture tracts, they enter the population, and then they got broken up by recombination, and then they don't recombine back together again. You can get some very simple analytical results for what is the distribution of the admixture tracts under very complex scenarios of integration happening over a number of years, then stopping and then starting again, and changes in population size, whatever you want. The math becomes very easy in terms of deriving analytical prediction. And this is some work I did together with my postdoc, John Poole, where we looked at the integration zone in Europe between two mouse species, Mus musculus domesticus and Mus musculus musculus, and we could estimate when they have started to interbreed with each other in this integration zone and when they stopped, and test whether they've stopped interbreeding and so on, test various parameters like that. Now this, I would say, is sort of a first step forward, but we're kind of not content with this because we made this assumption that these admixture tracts, they can't recombine back together again. And that's a very unrealistic assumption, particularly if you have a lot of admixture coming in. If they can recombine together again, then the demographic history of the populations matters a lot and things become much more complicated. We did next, and this is work by a student of mine, Mason Liang, was we developed a method for simulating pedigrees infinitely, a method that could simulate a pedigree infinitely far back in time, or samples from that without having to simulate the entire population. So we established a stochastic process that runs along the length of the chromosome that can sample ancestors in such a way we, in a realistic model of pedigrees and where we have, two sexes, male and females that have to mate and so on to produce offspring, we could then generate distributions of admixture tracts under this model very easily. And so these are some simulations under different assumptions about when admixture happened and also the admixture proportion. And what you see in black are the simulation results. So hopefully people also in the back of the room can see this, I'm not quite sure. But the y-axis here is logarithmic, and that means that if tract lengths truly follow a Markov process, waiting time should be exponentially distributed, and the black curves here should be straight lines. And you can see the many scenarios where that's not true, where you clearly get something that's very much not exponential. Okay, so you get some tract length distributions that are very, very different from what's predicted on our macro model. We also looked at various approximations to this, that people have been trying to do some various macro approximations, and one approximation here that's the most common approximation used many places, that's called the SMC approximation. We can see shows very, very, very poor. Another approximation that's called the SMC prime performs a little bit better. But the major take-home message is that tract lengths are not exponentially distributed. They don't follow a macro model. Okay. Another point that I didn't is that they tend to be very clustered in the genome. And why are they clustered in the genome? Well, you can imagine first you get a chromosome that comes in, and then it gets broken up. But before it gets broken up, genetic drift might have increased the frequency of that fragment in the population. And then after that, it gets broken up in little pieces. So get all these little pieces clustered in a certain area of the genome. So get this clustering of the tracts in the genome. They're not randomly distributed in the genome. You'll have clusters of admixture tracts. And that will tend to mislead the HMM methods, because they might stick together all these smaller tracts that are clustered in the region into one big tract. Okay. Deviation from the macro process occurs at many different timescales, and then also some other conclusions about which approximations may or may not be good to this. Okay. Next thing we want to do is a relative more theory of the admixture linkage disequilibrium. And I'm going to explain what that is in a second. I'm just going to mention that really much of the theory is outlined or comes from two papers, one old papers that really outline much of the mathematical formalism needed to deal with these issues. A paper by Bennett from 1952, another paper from Slackton from 1973. But there's a really, really old-fashioned stuff. But it's very, very, very powerful in this particular connection that can give us this 3-point admixture linkage disequilibrium. The admixture linkage disequilibrium, well, maybe I'll start with this slide. So you're hopefully familiar with normal linkage disequilibrium. If you do anything with SNPs or anything with genetic data, you should be familiar with linkage disequilibrium. So it's, as you will be aware of, it's the covariance between allele frequencies at two different points in the genome. So we can define this covariance function, which is the classical linkage disequilibrium, and defined in terms of this coefficient of linkage disequilibrium that's calculated as this covariance function. We used to look at admixture because we're looking at what's called the admixture linkage disequilibrium. Because of these tracts that come into the genome, it will increase the covariance. And that's called admixture linkage disequilibrium. And there are a couple of methods that have been published for doing that. There's a program called Alder and another one called Roloff that looks at the decay of linkage disequilibrium. So it's illustrated by these figures here. So you have distance in the genome here in terms of centimorgan on the x-axis. On the y-axis, you have a weighted form of this coefficient of linkage disequilibrium or covariance function in allele frequency along the genome. And people looking at how that decays. And if you have a lot of recent admixture, it will decay at a different rate than if you have a lot of old admixture. So if the mixture is old, you decay fast. And if that mixture is recent, it will decay slowly. So these programs, what they're trying to do is they're trying to figure out what is the rate of decay of this admixture linkage disequilibrium. Can that tell us about when admixture happens? Okay, the covariance function is still linkage disequilibrium. What we're interested in is extending this kind of way of looking at it to higher order correlations. So we want to look at, for example, 3-point linkage disequilibrium. The 3-point linkage disequilibrium can be defined as a 3-point covariance function between 3 points in the genome. So this is the mathematical definition is here, if you're not familiar with 3-point covariance. And that's in fact something that goes back to that 1952 paper by Bennett, where he does some things, has a similar definition there. Okay. To get that, now the 3-point covariance function, you just need to get the covariance in admixture proportions. So the covariance between an indicator function for whether an individual is admixed or not at a particular point in the genome. And that's what we need to calculate are things like this, the probability. So this is an indicator function for a haplotype being admixed, coming from the admixing population at position X in the genome, position Y, and position Z. So the joint probability that they're all admixed. So equal to 1, meaning that they're admixed. And we need something like this, the probability that you are in position Y and Z times the probability that you are in the third position, and so on. So these are the different probabilities you need to be able to calculate to be able to get that 3-point covariance function. So what we can do is we can set this up as a vector, and then we can have some matrices that operate on that vector that models the effect of gene flow coming in, the admixture, models the effect of genetic drift, we can also model things like mutation and other factors on that. This is, you have one matrix here that's migration from the other population, one matrix that has to do with the effect of genetic drift, and this is a matrix that has to do with recombination. We could also put in other things like mutation and selection, but to start we have recombination, genetic drift, and gene flow. It turns out that these matrices have a shared eigenvector. You can get a very simple analytical expression. Derive simple analytical expression for the 3-point covariance function now. Okay, so this is on a single migration pulse. That's, if you imagine the migration just having a single point in time, we can write down exactly what the 3-point covariance function should be in terms of the migration, proportion of migrants, the effective population sizes, recombination rates, and times since the migration happened. We can derive other expressions for more complicated models, but of course the equations are not then quite as simple as this. But okay, what can we use that for? We can, for example, use it to estimate times of admixture if there were 2 admixture events, or if admixture starts and then stops and so on. Because now we can look at this higher order linkage disequilibrium. So you can estimate that, you can provide estimates of parameters, more complicated models from this. You can do something, you can use the same kind of formalism, something else, which is variance in admixture proportions. Okay, so if you run a program like Admixture or Structure, if you're familiar with any of those programs, what you'll get out, the result is something like this. So each column here represents an individual, and then the color coding is, you're assuming that the 2 populations here, blue and red, and the proportion of that individual that's from the blue population and from the red population. So this is for some African American individuals, an estimate for each of the individual. Blue here represents African ancestry and red representing European ancestry. So you'll see in the literature literally thousands of plots like this. And what we're interested in, using the fact that there's a variance among individuals in these admixture proportions, and use that to try to estimate also parameters of the history of admixture between the populations. And how does that relate to the previous example I was giving? Well, to get this variance in admixture proportion, you need to be able to get the covariance between different points in the genome, and that we could get using that method I just described. So now what we can do is we can look at something like this, where you can put time. Imagine that there's been very recent admixture, and so some individuals are completely unadmixed, some have ancestry from the blue population and some from the red population. Now, as people in that population breed and keep breeding, you have some mixing that's going on, so that the variance among individuals becomes smaller and smaller and smaller. So, for example, after 500 generations, you might have something like this. And so what you can see is you have variance among populations. They will converge to different proportions because of the genetic drift. But the variance among individuals within population, that will go to 0. And this variance among individuals within population, we can use then to infer how long time since that mixture happened, using methods that are mathematically very simple when first we have derived the results that I described previously. We get the Taylor variance very easily, just using the 2-point covariance function. And if we use its mapping function for recombination, this is an expression that gives you, this is the variance that you would get in the of admixture fractions among individuals in a population. So we can again write down very, very, very simple analytical expression for this. Okay. And we can extend it in many ways to higher moments, more complicated models of admixture, and more complicated models of recombination, and for inference in various different ways. Because now we have a full mathematical model that can describe this process of admixture. So we think we have a mathematical description now of how admixture works in a population that we can use in many different settings, and we're hoping to use that also to improve models for inference of these admixture tracts. So, for example, using the models that 23andMe and other companies are using to do ancestry inference, I think that can be improved by having better mathematical models that better describe actually the process along the length of the chromosome. We can also, of course, use it for this kind of inferences, for some African American data. We have here a model where we're trying to estimate when did actually admixture between Europeans and Africans occur in the ancestry of African Americans in the United States. And the best fit we get is that admixture started about 11 generations ago, but largely stopped 2 generations ago. And that fits with various anthropological studies that argue that in the last few generations there have been much less admixture actually between African Americans and people with European ancestry than in previous generations. Okay. For that, but I want to make sure to point out some of the people that really been doing most of this work, particularly Mason Liang here, is the one that developed all. He's a student of mine, now former student, that developed all of the theory on admixture tracts I was talking about. Emilio Sanchez led the work on the Tibetans, a postdoc in my group, now a professor at UC Merced. Matteo Filmogali, another former postdoc in my group, led the work on the Inuits. Many other people have been involved in this research, and their names are listed up here. Thank you so much for listening to me. Okay, so you can do it many different ways. So in that particular study, the way we do it is use a statistic that's called the PBS statistic, which is you have 3 populations. You have at hand, and we have an outgroup, the Europeans. And then for each particular SNP, you calculate FST, which is something that measures difference in allele frequency. You make a mathematical transformation so it becomes linear in time under the assumption of genetic drift. So now you have a statistic pairwise between 2 populations that tell you something about the amount of time it will take genetic drift to create that difference in allele frequencies that you see. Now you have that between 3 pairs, and from that you can then puzzle out how much genetic drift, what's the amount of time that would require to create this much genetic drift, particularly on the Tibetan lineage. And then you're simply looking for SNPs where that time is very, very large, unrealistically large. And then to assign p-values, what you do, that's where things becomes very difficult, because to assign p-values, you can only do that under a particular demographic assumption. So now you have to try to estimate a joint demographic model for these populations, and then do parametric simulations to find the distribution of your test statistic. That's a little awkward, but unfortunately, there's nothing we can really do in terms of detecting selection that has any power, doesn't somehow depend on the demography. You always have to go through this painful process of trying to estimate demographic parameters of these populations, and then simulate new data to get p-values. How long time it takes to finish it? I mean, you mean how long time it has taken for it? Yeah. Yeah, sure. So we can estimate that. So we have mathematical models of how natural selection works, and we can estimate it. And what we can show is for the Tibetans, it seems to be relatively recent, 5, 6, 7,000 years maybe. For the Inuit, it seems to be much older. We're thinking actually as old as 20,000 years. And that's a bit surprising because people, the Inuit did not move into the Arctic before maybe a few thousand years ago. So we think this is actually much older selection from some of these Siberian populations that have lived in the near Arctic for much, much, much longer time. Well, some of them might be. So in the paper, when we published this back in 2010, the data for the Han Chinese was part of the 1,000 Genomes project. And the agreement we have on them was we could not comment on anything on their data. So we couldn't say anything about selection on the Han Chinese. We're not allowed to say that. But we could only say something about selection on the Tibetans. Now, those ones I happen to know, many of those ones are known genes that have to do with skin pigmentation. So if you look at some of the ones that show strongest evidence of a change in allele frequency, where the Han Chinese have a higher allele frequency in Tibetans, many of those are associated with lighter skin pigmentation. Yeah, I mean, we can show that they have, in terms of cholesterol, they have and fasting insulin levels and so on, all these endophenotypes. We can show that they are less protected than the other ones. And in fact, because of that mixture, so the Inuit population is highly admixed with Europeans, and so we have many individuals that live that have 89% Inuit background, but then don't have these mutations. And we can also look at phenotypes in those, so we can see there's a very strong effect where this mutation is protective. And the question is why you don't have it in other populations when it's so strongly protective generally in the Inuit, but presumably it's because of difference in diet traditionally. Yeah, so well, in terms, I can tell you a little bit about what we know. So we didn't do it in this study because we didn't have any exome. We didn't have access to samples that we could do exome sequencing. But we know the Tibetans who are most closely related to, which are Yi populations and so on in that southeastern corridor, in the Tibetan corridor there. So the Yis in several studies have been shown to be most closely related to Tibetans. In fact, people from India and Pakistan and so on are not particularly closely related to Tibetans. Tibetans are definitely formed in group together with all East Asians, particularly the Yis, some of these other populations are close, but Han Chinese are also relatively close, and one of the more closer populations.